Our next speaker will move us from, well, let's see, I guess you might say that there's one four-letter word that's very important in everything we're talking about today, and that's data. And we've heard about the electronic health record and other areas where we need data to inform policy and provide good patient care. And now we're going to turn to the science side, and we're going to begin with David Humes from the University of Michigan, who has worked in a variety of areas: acute renal failure, stem cell biology, and tissue engineering. And I know he'll tell us a bit about some new dialysis technology, as well as whatever else he has in mind. But as I say, now we've gone from some of the policy areas to look at some of the science areas. Of course, they have policy implications as well. So, David, please.
Thank you. It's always good for a country doctor to come to the big city. A lot of fun here. But it's grand. It's an eclectic meeting. I'm honored to be invited to talk about some of the science and new technology that may help patients as well as save costs. In the typical fashion as mandated, I should do disclosures because I have several biotech companies that have spun out of the university and have relationships to develop and commercialize this technology in order to move it from the bench to the bedside. I'm a traditional translational scientist, an MD, and it's a rocky road, but it's a fulfilling road, and I'm going to go through some of our technologies and how they developed and what the implications and impact it may have on clinical care. You don't have to look very far in nephrology to look for the unmet medical needs. Acute kidney injury, acute renal failure has mortality rates still exceeding 50%, especially in the ICU where mortality rates go up to 70%. Of note, this mortality rate really has not changed over the last 30 or 40 years. End-stage renal disease is life-sustaining, but as all of us know, it is a debilitating disease process. Average life expectancy is 5 years. It's debilitating enough that many of our patients withdraw from dialysis, a form of suicide. So we have to do better. And I go back in the literature, and nothing's really new, but I think Ben Burton, who was the director of the chronic uremia program of the NIH NIDDK program, stated way back in 1976, after about 5 to 10 years of dialytic therapy beginning to develop, he stated maintenance dialysis on the whole is non-physiological, and because it can be justified only because of the finiteness of its alternative, life or death. While industry appears to be faring well, and even better now, research-minded professionals, and that's all of us here in this room, are beginning to recognize that we face a distinct danger that dialysis may become institutionalized. Oh, I think that's become true. Satisfied with what we have wrought in this field, we will pile small improvements on top of other minor advances in dialysis technology. Ultimately, the technology of dialysis therapy will become entrapped in its own net for failure to break out in new directions. And I think it's our role as scientists and doctors to really break out of that net and try looking at where the technologies may be improved to better care for our patients and meet their unmet medical need. When you look at why patients die with either acute kidney failure or end-stage renal disease, what is remarkable is that it's not because of lack of small solute clearance, but a systemic inflammatory response state that is either acute or an end-stage renal disease that is chronic, leading to accelerated atherosclerosis and a susceptibility to infection. If you look at it in its broad form, we have evidence developing in our laboratory that renal tubule cells are critical with regards to playing an immunomodulatory role for the body as a whole. In acute renal failure on the left, there's a diminution in host response. There's an inability to modulate the pro-inflammatory state, all leading to an acute pro-inflammatory program, multiple organ failure, and death. In chronic end-stage renal disease, there's a persistent chronic inflammation as measured by CRP or IL-6, leading to accelerated atherosclerosis, malnutrition, and early mortality. If you look at the epidemiology of this, a 20-year-old patient developing end-stage renal disease becomes, from a risk factor, an 80-year-old with regards to the acute mortality from cardiovascular processes. So the kidney is more than a clearance organ. It's more than an excretory organ. And the cell biology of the tissue is critical with regards to its immunologic, hormonal, synthetic functions. We have been focused for the last 10 to 15 years on acute renal failure. It's going to be detailed more in terms of the science in the next talk. But we recognized from the pathobiology that acute renal failure is really due to injury, necrosis, and apoptosis of the lining renal epithelial cells. The kidney is unique that with acute organ dysfunction and failure, there's a regenerative, resilient subpopulation of cells that have the ability to regenerate with time so that you can recover 90 to 95% of the time to baseline renal function. So that over a decade ago, we began to look for, in the adult mammalian kidney, cells that had the ability to expand in a progenitor or stem cell fashion. Being able to identify them and isolating them from various different mammalian adult kidneys, including humans, we obtain human adult kidneys on transplantation discards that cannot be used for transplantation, either because of anatomical defects or greater than 30% sclerosis, especially in the glomeruli. We were able to develop a renal tubule assist device that was called the RAD that had cells within the lumens of a conventional hemofilter so that we can put a nephronal extracorporeal device in series for treatment of patients with acute or chronic renal insufficiency. So we have a synthetic hemofilter acting like a glomerulus. Filtrate is then driven into the internal interstices where lining cells are available, post-filtered blood with a step of oncotic pressure driving transport and reclamation, and synthetic derivatives being produced after signals from the ultrafiltrate to be returned to the patient. So now we have a bioartificial kidney, both with a filter as well as a metabolic tubular element. This is schematized here, in which the second element, the renal tubule assist device, have differentiated renal epithelial cells that are immunoprotected by a scaffold-based membrane and an ability to move into treatment paradigms with this type of setup. After a large amount of preclinical studies, FDA approval, identification of human sources, and expansion and building a GMP, good manufacturing process, renal tubule assist device, we went into clinical studies. We went through a phase 1-2 study, a phase 2 study design in which ICU patients were identified in multiple medical centers with acute renal failure and multi-organ failure. They were randomized to 2 cell therapy treatment and 1 conventional treatment with continuous veno-venous hemofiltration. It was open label, and we were allowed for 72 hours of therapy, mainly because the blood tubing and the hemofilter that we based our cell therapy product on was only FDA approved for 72 hours of continuous use. We demonstrated in 58 patients that we had a 50% relative improvement in survivability at 28 days, 67% with cell therapy, 39% in conventional therapy, and we went on to a phase 2B trial. This is the Kaplan-Meier survival curve in that first phase 2 trial, demonstrating with an improvement in the dotted line of survivability of patients treated with cell therapy versus a conventional control group in solid lines. We achieved a Cox proportional hazard ratio of 0.49, suggesting that about 50% of that observed in CVVH conventional therapy was improved in terms of the risk of death. As we began to develop a commercialization process for a phase 2B study before a phase 3 pivotal study for FDA approval, we identified that we had problems with cell expansion and device fabrication in terms of how we would store it and distribute it on a broad-based basis. Because of these complexities, we decided to step back, understand the mechanism, as well as improve our commercialization manufacturing process. And over the last 2 or 3 years, we've improved the device from a standard form of hemofilter to what we call a bioartificial renal epithelial cell system that is much more compact, containing 10 times more cells. This is based upon a scaffold of carbon niobium discs, in which it's a coral reef type of process that we can drive ultrafiltrate through 20 of these discs for a 10 to the 8th number of cells, so that we had an ability to use these cells grown along the trabeculated systems. These are DAPI stains. Each of the blue dots is a nucleus. This forms a confluent monolayer. And we have demonstrated with our bioengineers that you can scale up this device with cells, put it in liquid nitrogen for up to 4 months at minus 140 degrees, and rapidly thaw it within 30 minutes with less than 5% of the cells. So we have solved the distribution and storage process, plus we have improved our stem cell progenitor cell expansion from adult kidneys so that we can make not 5 to 10 devices, but over 100,000 devices from a single kidney. We have improved the cell sourcing so that from a single kidney we can expand not to 10 to the 10th cells, but 10 to the 13th cells, so that we've improved the ability to expand these types of manufactured products. So we've, over the last several years, we've miniaturized a cryopreservable renal cell therapy device. The Brex has been fabricated from a variety of different types of adult mammalian kidneys. They contains 10 to the 8 to 10 to the 9 cells. The Brex can be cryopreserved, and it has been demonstrated with efficacy in a porcine model of acute septic shock, so that we're ready to go into the clinic again with a much better manufactured process. But good science makes even better and more simple products. We have also began to develop a preclinical animal model of a sheep that is uremic, and currently testing a wearable regenerating dialysis system using sorbent-based technology, and the Brex as a biologic component of this type of technology. We've been able to demonstrate at 25 mLs per minute with recycling peritoneal fluid. We can maintain these cells up to 4 weeks, which is the limit that we're allowed to test the animals with. And we can regenerate so that you can now, with about 4 liters of fluid, be able to have small solute clearance with the recycling, regenerating dialysis system with peritoneal dialysis, and a nutrient and oxygen source to maintain biologic products and a biologic component of the kidney, so that you now can conceive of a wearable bioartificial kidney for small solute clearance as well as biologic components. But in the meantime, we've also been exploring how the cells mechanistically change in acute renal failure, acute injury, acute kidney injury type of state. we began to look at how the cells were able to perturb a uremic and inflamed state of acute renal failure and how it may modulate back to health a patient that has a 50 to 70% mortality rate. When we began to examine the cartridges after clinical use, we found that there were cells attached to the outside of the hollow fibers. Remember, the tubule cells were inside the hollow fiber. The fibers were used as an immunoprotective scaffold, but we were surprised that we found large amounts of cells, and upon evaluation of those cells that were adhered to the membrane in patients treated with acute kidney injury and multi-organ failure, it was predominated 80% by neutrophils and 20% monocytes. And it was only the activated neutrophils and monocytes that were being sequestered in this type of membrane system. We also demonstrated that upon sequestration, the cell products derived by the renal epithelial cells basically immunomodulated them, detached these cells, moved them to quick apoptosis, so that basically it was immunomodulating the circulating state of immunocompetent innate immunologic responses in a diseased patient continuously with regards to sequestering the cells and deactivating them so that you have a reduction of the excessive pro-inflammatory state of multi-organ failure. So we began to develop a biomimetic membrane device that would sequester the cells and be able to inhibit their immunologic effects by using a well-developed protocol of citrate anticoagulation that lower ionized calcium that would inhibit and deactivate the sequestered leukocytes. So the idea was, rather than using cells, understanding the science of how the cells were influenced a systemic inflammatory disease state, we would take a patient with acute renal failure on continuous hemofiltration in which there was ultrafiltrate. We would drive it down now a non-cell containing fiber that had biomimetic properties to sequester this, and with citrate anticoagulation with the low ionized calcium component, we would deactivate the cells using a pharmacologic substance rather than a biologic cell component to do the same thing. We did a series of approaches with regards to suggesting, non-conventionally, rather than focusing on the cytokine production in the systemic inflammatory response state, we did a proximal effect to alter the leukocyte or the cellular component that was producing a large amount of the cytokines, hoping that if we inhibit leukocyte activation, immunomodulate downward, that we would have a major effect on microvascular dysfunction, the capillary leak and poor tissue perfusion, as well as leukocyte tissue infiltration, and minimize the ischemic and toxic tissue injury that was evolving after acute kidney injury, leading to a spiral downward of multi-organ dysfunction and death. So it was a simple system in which we have standard dialysis pump systems with a conventional CVVH hemofilter setup, now put in series with a biomimetic selective cytophoretic device system to sequester leukocytes, and the low ionized calcium environment would deactivate and immunomodulate down. In vitro experiments have demonstrated by shear flow video microscopy that only activated leukocytes bind to this membrane. In vitro blood systems have demonstrated that low ionized calcium of the mode that we use for citrate anticoagulation deactivates leukocytes and monocytes in terms of cytokine and preformed proteolytic enzyme release. We moved into large animals of septic shock and demonstrate that we can immunomodulate, improve cardiovascular performance, protect against renal dysfunction and pulmonary leukocyte infiltration and pulmonary dysfunction, and increase survival times, so that we moved into a clinical data set. This is the first retrospective analysis of some of our older data using now non-cell cartridges, an SCD device, in an extracorporeal circuit with citrate and with heparin protocols. This is a typical historical control curve of a patient in the ICU achieving 50% mortality at 28 days and progressing with up to 6 months of follow-up, a progressive survival or mortality progression. With citrate and 2 devices, we were able to show that we had a significant effect. These are 12 patients in the citrate and 12 patients in the heparinized protocol with the selective cytopheretic device, that we only had a 75% survival rate that persisted to 67%, suggesting a strong clinical signal. We then went to China to further test a phase 1/2 derivative with an improved biomimetic membrane, and we allowed 72 hours of continuous CVVH with the SCD treatment, and after 72 hours, we replaced the patients on intermittent dialysis until renal recovery or death. 10 patients enrolled, met protocol, 7 of 10 survived. 2 patient deaths was a DNR decision. Concurrent controls had a 60 to 70% mortality rate. When we introduced an advanced selective cytopheretic biomimetic membrane in the last 5 patients treated, 5 of the 5 recovered renal function within 72 hours. The typical recovery of renal function takes 10 to 14 days if the patient survives. 4 or 5 survived, 28 days are discharged. The 1 death was because of family's request to withdraw even though the patient was improving but needed ventilatory support for recurrent pneumonia. So basically, we have approached the FDA and have gotten an IDE approval, and we are now in a multi-center single treatment mode evaluation as a pilot study. And the first 6 patients treated, which was started last month, demonstrate a similar type of early renal recovery, a high survival rate, and mimics the China experience. We're allowed up to 35 patients in the pilot protocol, and then we'll be allowed to do a pivotal IDE FDA-approved protocol with concurrent conventional therapy to compare the efficacy of this type of approach. We have also completed a 15-patient study in ESRD, and we have demonstrated that a single 4-hour treatment with the SCD in a conventional hemodialysis unit with citrate anticoagulation lowers pro-inflammatory markers, including IL-6, for up to 4 weeks after a single 4-hour therapy. It appears that we are pulling activated monocytes from the patients and then deactivating them and probably removing them from the systemic circulation. Since monocytes have greater than a 7-day half-life, we may be affecting the overall chronic pro-inflammatory state by pulling out activated monocytes that are the cause of the accelerated atherosclerosis process and the pro-inflammatory chronic state in ESRD. That's going to be a much harder evaluation with regards to the impact on patient survival because it's going to require hundreds of patients over 3 to 4 years of longitudinal study, but has some suggested major improvement with regards to altering a chronic pro-inflammatory state. So basically in the last 20 minutes, I've demonstrated how looking at the kidney as more than a clearance organ, looking at how you might be able to expand stem cells to make a cell therapy derivative. You can develop commercial principles to make it a good cell source and commercially viable with good storage and manufacturing processes, and then an ability to use this with regards to a wearable bioartificial kidney for end-stage renal disease applications, understanding the science of the cells and how the cells may impact an inflammatory disease process, devising a biomimetic membrane that can duplicate that cell therapy with another pharmacologic agent, and performing translational medicine at the bedside to prove its efficacy and safety characteristics through an approval process through the FDA. So this, in summary, is about 12 years of work with our group, but it's a traditional, true translational medicine in which you move from the bench to the bedside. So, lessons that I have learned, especially for the younger individuals in the audience in translational medicine, it's certainly not easy. You trust your intuition. We were told as a group that we were barking up the wrong tree, that there was no way we were going to be able to achieve either a cell therapy device or a biomimetic membrane that can change a systemic inflammatory disease process. Conventional wisdom is not wise. Disappointments and setbacks will occur. There are highs and lows that I don't think you can even imagine. But if you follow the science dedicated and you look at the data, you will come into understanding a disease process and a better way of approaching it. Certainly no one will do anything if they knew how it would turn out. And this is research. We don't know where we're going. And doing clinical research gives you a better understanding of the pathophysiology and how you may alter a therapeutic approach. And most importantly, we are driven with passion, persistence, and perspiration because we hope we may help the patients that we treat on a daily basis that need our care and improved technology. Thank you for your attention.